Establishing the segments in Drosophila. Morphogens initiate differential gene expression.
As we recall in our discussion in Chapter 3, different maternal transcription factors and signaling pathways have an impact on the establishment of body access of our fruit fly. The bicoid, nanos, dorsal, and the localization of the torsal-like receptor all have an impact to the establishment of the body axis of a fruit fly. All demonstrate the foundation of pattern organization, which is the asymmetric cell division and regulated signaling. The complexity of the mechanism of patterning in a Drosophila body, which has 14 segments, leads scientists to question, how did it come about? The answer is known, and it is one of the foundation of developmental biology. The series of complex networks of regulated pathways of transcription factors have been the key answer to the segmentation of Drosophila body, which has been come to be called epigenesis. These have been further elaborated in the genetic screen study by Nusslein and Volhard with the use of mutant fruit flies. It has been postulated that maternal transcription factors activate gap genes, which are expressed during the 9th to 14th cleavage cycles. These gap genes then produce transcription factors that lead to the formation of unstable mRNA. Then, the two-step process in which a pair rule gene encodes transcription factors that lay down the 7 broad stripes. Then, a segment polarity gene that divides these 7 broad stripes into 2, producing 14 segments with identifiable antero-posterior axis. Then, a homeotic selector gene which gives each 14 segments its separate identities. This summarizes the postulation in Nusslein and Volhard's experiment. Gap genes such as the tailless, Hackbian, giant, hunchback, Krupel, and NERPs interact with one another and with the products of the terminal axis system to establish the 7 broad stripes. These stripe domains then set up the condition for the regulation of the pair rule gene. Gap genes' expressions are highly regulated by the concentration of other transcribed gap genes, which limit their transcription and in turn their expression of their function.
Pair rule genes activated by gap genes create 7 repetitive stripes. Gap genes and bicoids. The activation of gap genes were caused by maternally deposited gradients of transcription factors. The transcription factors encoded by gap genes and bicoid help regulate the promoters of genes that establish the anterior-posterior sequence of 7 circumferential stripes, which is an important step because that comprise the basic body plan of Drosophila. 8 pair rule genes. The main function of 8 pair rule genes encode transcription factors. Primary pair rule genes are 8. 3 among the 8 are Runt, Harry, and Eve. So I will be discussing to you Eve because it is the most studied among the 8 of them. So one of the primary pair rule genes is Eve. As what you can see in the slide, it is first expressed in a wide region covering most of the embryo. Then, a single broad fuzzy band appears, as what you can see, which narrows and sharpens to become the first of 7 stripes. Then remaining 6 stripes then appear, pretty much at the same time. Gradually, they become more defined, attaining sharper and sharper borders, as what you can see in the slide. Primary and secondary pair rule genes expressed in 7 stripes of activity, so they have their own process, but it's still the same. In addition, the borders of their expression domains do not exactly align. Instead, the domains overlap, thereby creating complex repeating patterns of expression.
Pair rule genes have complex regulatory regions. Now you might wonder how this striped expression is attained. Well, each pair rule gene has a large, complex control region that has dozens of different sites with varying gaps, and this is where maternal gene transcription factors are bound. The binding of a given factor may activate or repress transcription. The transcription products of the regulatory genes interact with either an enhancer or suppressor region on the target pair rule genes. The conditions of the expression is set by the exact pattern of transcription factors that are bounded to the control region. Stripes are specified individually by the upstream gap and maternal genes acting directly on the DNA regulatory regions that control even-skipped expression. The primary pair rule gene has clusters of sites that activate expression when they are occupied. There could be 7 clusters per pair rule gene, and each respond to a different combination of gap gene transcription factors. Because of this, the pair rule gene is turned on at different locations within the embryo. Turn off gene expression in cells take place because suppressive sites are scattered throughout the regulatory region. Now we have both turned on and turned off gene expressions, thus forming the stripes. These stripes are enhanced when interactions occur, which vary with the 7 pair rule genes. An example of this is the activation of the EVE stripes as illustrated in the picture being shown. The gene EVE is regulated by both activating and inhibitory interactions.
The gene Eve is regulated by both activating and inhibitory interactions. And let us take a look for more details of pair rule genes to discern how the stripe arrangement develops. In this figure, it shows the binding sites on Eve for several proteins. The sites are in a region 800-1500 base pair upstream from the transcription starting point. In the figure A, we show the relative binding positions for the 4 proteins, and in B is the exact binding sites of regions of 2 different factors can bind to a particular gene. The positively acting factors and negatively acting factors can compete for certain sites. In general, activating and inhibitory sites are often adjacent or overlapping, and there are often multiple copies of the same site close to one another. The function of regulatory modules for several Eve stripes. In this figure, the reporter constructs are used to study the function of regulatory modules. They link to a reporter gene and placed in Drosophila tissue culture cells to see whether the artificial regulatory elements work or not. Or reporters can be introduced into embryo which was modified by genetic manipulations. In this slide shows an example of an experiment where the E promoter for stripes 1, 5, or 1 plus 5 together fused to the coding region for lac Z, where the A is the reporter, B is the enhancer for stripe 1 and 5, C is when the reporter constructs for stripes 1 and 5 in a null mutant of hunchback, and D is when the same construct is introduced into a proper stripe 5.
Expression of the segment polarity genes provides permanent markers for parasegmental boundaries. Before we consider how the identical parasegments come to differentiate along separate pathways, we should mention that many of these genes are only transiently expressed. However, expression of the engrilled and the other segment polarity genes provide more or less permanent markers of the segmental boundaries. So how is continual expression of these genes maintained? The regulatory regions of engrilled is huge and complex, which involves both upstream and downstream DNA, and the analysis of its organization is still incomplete. Nonetheless, we do know that engrilled has an autoregulatory region that participates in a positive feedback loop. This loop starts with the engrilled protein that acts back on engrilled by binding to a feedback enhancer that keeps the expression of the gene turned on. So there is a second important but complex way that continual expression of engrailed is maintained. It is the intercellular communication that involves the wingless expressing cell and the engrailed expressing cell. So the second way of the continual expression of engrailed gene comes by a ligand receptor interaction with a neighboring cell that expresses the wingless gene. So the engrailed expressing cell not only stimulates its own engrailed genes, it also stimulates hedgehog to synthesize the ligand hedgehog, which acts on the neighboring wingless expressing cell. So in this cell, a pathway involving the genes, which are the patch and smoothened genes, suppresses the wingless gene. So the hedgehog inhibits this patch smoothened pathway, allowing the ligand wingless to be secreted. So the wingless gene is subsequently received by the engrailed expressing cell, where wingless is a positive signal for the engrailed expression. Thus, the loop between engrailed expressing and the wingless expressing cells is maintained, each reinforcing its neighbor to do its own separate thing. So this engrailed and wingless signaling pathways turns out to be involved in many developmental pathways.